Bismillahirrahmanirrahim. Assalamu alaikum and welcome all of you uh, for celebrating Women in Science Day at IWD Forum.
कैमरा तो नहीं ऑन है
Start this.
लाउड मीटिंग ये देखो
Participants, kindly mute your mic.
ये क्या ये वाला अच्छा 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 मैं वो वाला ज्वाइन करना चाहती हूँ क्या है तो मैं सुबह साइनिंग की थी नहीं तो साइनिंग पहले करो ना कहाँ से करूँ
Am I audible? Can anyone tell, please?
Yes, you are.
Okay. So let me start today's STEM Cafe featuring women in science, celebrated globally around the world, that is on 8th March. So with a few words about the role of women in science, we have just a report to share here at IWD Forum that is according to UNESCO Science Report towards 2030. This will be the women standing across South Asia, and Sri Lankan women will be leading with 41%, and followed by the Pakistani women, that is 36% in their role in STEM education. So the role of women in science has evolved significantly over time, but historically, women have often been underrepresented and faced barrier to entry and advancement in the scientific field. Despite these challenges, many women have made scientific contribution to STEM fields throughout the history. Today, efforts are being made to promote gender equity in STEM and support women in pursuing and thriving in scientific careers. So despite the cultural boundaries in Pakistan, women had made the notable contribution to science. And despite these challenges, such as the limited access to the educational opportunities and societal expectations, many Pakistani women had pursued their scientific careers. IWD Pakistan is a forum that believes in appreciating, encouraging and highlighting the role of such wonderful ladies who, despite all the challenges, are contributing in STEM towards the betterment of the globe. So we at the IWD Forum, and I on the behalf of whole IWD team, appreciate all the world globally, all the women globally who are contributing in the STEM field and playing their role for the betterment of this globe in medicine, in science, in technology, in engineering and mathematics, and whatever the role they can play. So hats off to all you ladies. Here I am showing you the screen with IWD Pakistan social media links where you can be the part of this forum, and you can also highlight the role of any such lady if you are among them and you are playing your role towards the betterment of the globe, contributing in STEM fields. You are most welcome to be the part of this. You can contact IWD team at any of these social media links. You can be the part of this. With these words, I will move toward today's celebrating International Women's Day speakers. Here today we have two speakers, Dr. Raza Ashraf and Dr. Bushra Nisar Khan. With a brief introduction of these wonderful ladies, we will move ahead towards their session, and we will like to know about their journey and their scientific contributions. So Dr. Raza Ashraf, she is working as an assistant professor in Department of Biochemistry, the Islamia University of Bahawalpur. She has done her PhD in biochemistry from University of the Punjab, Lahore. She has done her postdoc from Department of Biochemistry and Biophysics, Stockholm University, Sweden. Currently, she is doing postdoc from Faculty of Chemistry, University of Duisburg, Germany. She has worked on various research projects and currently working on different projects as well. We will like to know the details from her shortly. I would like to introduce the second speaker for today's session, Dr. Bushra Nisar Khan. She is working as an assistant professor in Institute of Zoology, University of the Punjab, Lahore. She has teaching and research experience of over 11 years. She is an author of 50 research articles and 2 books. Her area of research expertise are biodiversity conservation and natural resource management. She is member of Pakistan Biological Society, Pakistan Fisheries Society, South Asian Association for Regional Cooperation, Zoological Society of Pakistan, and Zoological Association of America. We are really glad to have both of you here at IWD Forum, and I would like Dr. Raza to now please continue with her talk. Let me see if you can share your screen or not. Can you share your screen?
No, it's been disabled by the host. I think you have to allow me for sharing the screen.
Kindly try it again.
Yeah, now I can share. It's fine, I think.
Yes.
Okay. So, hello, good morning. Assalamu alaikum, everyone. I'm Dr. Raza Ashraf, and right now I am in Germany and doing postdoc. But first, I will like to introduce myself from where I really started in biochemistry. So, like everyone doing FSC and dreaming to be a doctor, like medical doctor, I was also one of them. And later on, but I could not make it in MBBS, and then I got admission in pharmacy, but I was not really happy with the pharmacy. And I also applied in Institute of Biochemistry and Biotechnology for BS in biochemistry. And unfortunately, I was informed that I did not get admission. So I was a bit disheartened that how it could be that I could not get admission even in biochemistry. It's impossible. But later, somehow, because I was far away from Lahore, I could not follow it, and I got admission letter from pharmacy department. I came here in university, like really not happy. And the people who are from Punjab University, they knew that when you take admission, you have to take some medical test from the University Health Center. So I came from old campus to new campus just for the medical test, but then I talked to my father that I just want to have a look on the admission thing, that why I could not make. And when we went to biochemistry department, I came to know that my merit order was third, and I was informed wrongly that I did not get admission. And then at that time, Professor Dr. Wahid was HOD of the department. And then we went to him, we talked to him, and he said, no, now it's not possible. And then we said, maybe with late fees. And he said, oh no, I will make it. Just I need admission here. I don't want to go in pharmacy. So I got admission in biochemistry. It was 2 weeks. I was stuck with presentations. I was stuck with assignments because sometimes if you don't know the task or the hurdle, you just go for it. Because I at that time I did not know that the marks of midterm are included in the final term. It was a good journey. And then I did my MS from the same department. And then I had a job in University of Islamabad. Now it's called Medina University of Islamabad. At that time, it was called Medina University of Islamabad. Now it's called University of Islamabad. But I had job there for just 8 to 9 months because then I got admission in School of Biological Sciences, Punjab University, and I came back to Lahore. And then I started my PhD. And that was a bit difficult journey for me because in first 3 years of my PhD, I had so many experiment, but I did not had any result. But somehow, suddenly I got some results, and then I went to Japan initially for 6 months by HEC. But there again, I had so many problems in my experimental work, and then I decided to extend it up to 1 year. Then I came back and I did my defense. Everything went well, and then I started job in Minhaj University and as a visiting in Punjab University and also in Education University. Like I was teaching in three universities at the same time, but after 6 months or an year, I just focused on Minhaj University. And then I got job in Islamia University, Bahawalpur, and I went there. At that time, I had two options. Either to like it, I was trying to get postdoc and I was not really getting any good answer, but suddenly I got postdoc offer and job offer at the same time. And then I went for the job because at the end I really wanted to stay in Pakistan. Then after a year, I went to Sweden for a short postdoc, then came back to Pakistan. Then now I am in Germany again for postdoc, and I hope so I'll be back in Pakistan soon. So this was my brief introduction. So, all those students who are doing BS, MS, or PhD, so they should never lose heart because of their bad results. It's part of the PhD. So now I come to my research work. I am working on multiple projects. One of them is metabolic pathways in archaea, and other one is formation of nanocages from ferritin proteins and encapsulants, and one is solubilization of protein. But today we will talk about the beta oxidation in Pyrobacillum caladifontis. Actually, this topic really fascinates me because of the idea that it could be the clue to the last common ancestor. So first we will go that how this beta oxidation could be the last clue to the last common ancestor. Actually, I think every one of us know that we have the cellular structure in eukaryotes that is bound by lipid bilayer. Like it's considered as a universal in all prokaryotes and the eukaryotes. And fatty acids are considered as one of the most important component of the cell, being formation of the boundaries. But in in archaea, the situation is a bit different from the situation in eukarya and prokaryotes. Because if you see here that in, I think you can see my wait, point. I should, yeah. So, okay, here. Here you can see that we have this figure for the standard lipids that are part of the lipid bilayer, that we have fatty acids and then ester bond and then glycerol. But the difference between the lipids of archaea and bacteria and eukarya is that if you see that here we have ester bond, but here we have ether bond. This is one difference. And the other difference is in the fatty acids. Here is the regular fatty acid chain, but here the chain is consists of isoprenide units. So because this is most important function of the lipids in the cell, So in archaea, it has been considered that if there is no fatty acids in the membrane, so there might not be any fatty acid metabolic pathways in the archaea, because their genome are very compact, and there could not be any additional gene which don't require their function. So everyone is considering, okay, there should not be any beta oxidation pathway. So if there is no beta oxidation pathway, there should not be any beta oxidation genes as well. Okay, but and now what is last universal common ancestor? So this is a very general diagram which showing the three domains of life, that bacteria, eukarya, and archaea. And here comes the last universal common ancestor, which could give rise to bacteria, archaea, and then it's considered that because of the overlapping of bacteria and archaea, there is the formation of this eukaryotes. And what would be the common ancestor which have the properties of both bacteria and archaea, which would have the genes which are specified for bacteria or specified for the archaea, and both are functional in that last universal common ancestor. So if an organism does not need beta oxidation pathway, and it should not have beta oxidation gene. If an organism does not need isoprenoid synthesis pathway, it should not have the genes for the isoprenoid synthesis. Then this is one of the modern domain of life diagram. Here the archaea has been divided into further groups, and there come another ancestor which could give rise to archaea and eukarya, and initially it divided into bacteria and eukarya archaea combined way. So we were focused on the archaea. Archaea are mostly extremophiles which can thrive on the very high temperature like 100 degrees centigrade, 110 degrees centigrade, or very extreme salt conditions, very extreme pH conditions, either at acidic, extreme acidic like 2 and 3, or extreme basic conditions. So they have very different properties which can make them to have a stable life at those extreme conditions. So actually, initially, my source of attraction toward archaea was that their extreme conditions, what make them to strive at very extreme conditions where other life cannot strive. So initially, I was the 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 source that I selected was Pyrobacterium calicifontis, and initially I was interested to clone some of uh, the enzymes which can play a role in the industrial side, because in industries we need very high temperatures and we need very high pH, and generally enzymes from the bacteria or eukarya, they cannot handle those situations. So if we talk about the Pyrobacterium calicifontis, it's an archaean which has been isolated from the hot springs in the Philippines. And interesting thing is that the optimum temperature for its growth is 90 to 95 degrees centigrade. And pH is just neutral pH and with aerobic conditions. And the doubling time is around 6 hours. Cells are rod-shaped. Uh, so my first target was to get some alcohol dehydrogenase with very stable condition. So there was a gene which was annotated as 1309, the gene annotation, which was alcohol dehydrogenase. I was working on this alcohol dehydrogenase, but I could not get very promising result for this alcohol dehydrogenase. And this alcohol dehydrogenase was behaving more like with the aldehyde substrates as compared to the alcohol substrates. So then we had a look that where what is the genomic location of this alcohol dehydrogenase in the genome of Pyrobacterium calicifontis, and then we really found a situation that was bit interesting, that this alcohol dehydrogenase was in the what we say in the genomic neighborhood of the enzymes which were really participating in beta, like we expect them to participate in the beta oxidation, and they were making an operon. So it was expected that they were working with the interaction of each other, and so there must be some pathway which really have function of these enzymes. Here I have highlighted with the color code that this is an hydratase. I can further explain in the metabolic pathway diagram what is the function. And then there is esterase, then there is acyl CoA domain dehydrogenase protein, then there is thiolase and DUF protein, domain of unidentified protein, alcohol dehydrogenase, then oxoacyl ACP reductase. And there was not one operon, but there was two operons with the similar genomic arrangement. So we were expecting that there must be some beta oxidation pathway as well as isoprenite synthesis pathway, because it was Archean, its membrane was isoprenite based, so definitely isoprenite biosynthesis pathway was working. But for the beta oxidation, we have to look into the metabolic environment and metabolic processes of the Pyrobacterium caldifontis. So if we talk about the beta oxidation pathway, it's about the breakdown of fatty acids, or in reverse, it could be the synthesis of fatty acids. Here, I am giving this diagram in the reverse order that starts with the thiolase here, which can combine two acetyl-CoAs to make the acetoacetyl-CoA. And then acetoacetyl-CoA reductase, which is 1312, can change its acetoacetyl-CoA 3-hydroxybutyryl-CoA. And this DUF protein, it might have two functions. One could be the conversion of this 3-hydroxybutyryl-CoA to S3-hydroxybutyryl-CoA to R3-hydroxybutyryl-CoA or R2S. And then this crotonase, which creates a double bond here by dehydration and converts this 3-hydroxybutyryl-CoA to crotonyl-CoA. And at the end, this crotonyl-CoA can be changed into butyryl-CoA by acyl-CoA dehydrogenase. And this can also go in reverse if we start from the down to upwards, that there is fatty acid and it can be changed into crotonyl-CoA by acyl-CoA dehydrogenase, then 3-hydroxybutyryl-CoA, then acetyl-CoA, then breakdown of one acetyl group from the fatty acids, which is the standard fatty acid metabolic pathway for energy production. Okay, but up to here, I have mentioned these 4 genes. The thing is that there were 2 more genes, and that was esterase and alcohol dehydrogenase. So we were expecting that this esterase can change this butyryl-CoA into butyraldehyde by breaking this CoA, and then this alcohol dehydrogenase can go for its preferred activity to convert this butyryl-CoA to butanol. And with this butanol can be used as the biobutanol. So we were more interested with beta oxidation pathway, but we were also interested either it can lead to uh, the pathway that can be involved in the biobutanol synthesis. So here are the genes to operons, but right now I will talk about just one operon. Okay, we have the genes, we have the expected activities, but how we will do it? Because it was very difficult to purify a protein from the organism because it was extremophile, its bubbling rate is very slow, and protein purification without any tag was difficult. So what we did, we went for the cloning options, that we will clone each gene in E. coli, which is an easy host, and we can easily purify protein. So first thing was to grow Pyrobacterium caldifontis. And then the second thing was to isolate genomic DNA. And then we designed primers and we did PCR for each gene separately. And then we went for the cloning and expression in E. coli. And we had transformation, overexpression. Then we just disrupted the cells to bring these proteins out. Here I just showed one protein, but this was the same protocol for all of them. And then we went for the characterization, that at what conditions these protein could work. What could be the substrate concentration, optimum substrate concentration, optimum temperature, optimum pH. So we know that, okay, at under what condition we can get a standard product from the substrate. So I'm not going into details of each enzyme, that what is the optimum conditions for each enzyme, but I am just going into the conversion of substrate to product. Okay, the first enzyme that we successfully cloned was crotonase, and its activity is known as to convert the crotonyl CoA to 3-hydroxybutyryl CoA and 3-hydroxybutyryl CoA to crotonyl CoA in the reverse reaction. Here I have highlighted this. We analyzed this reaction with HPLC. This is a bit bad chromatogram, but it's okay. So here we have in green line, sorry, first we talk about the standards. In the red line, it's crotonyl CoA, standard crotonyl CoA. In the here it's bit, what can I say, blue line, that is standard 3-hydroxy butyryl CoA. Actually, it's black. And then we have two reactions. One was the control reaction where we have protonyl CoA, but we don't have enzyme. And then in the other, we have protonyl CoA with the enzyme. And you can see that in the green line, there was no peak at the standard 3-hydroxybutyryl CoA. And in the blue line, we have the consumption of this crotonyl CoA. The peak size decreased, and here is a small peak which appeared at the 3-hydroxybutyryl CoA. So we were sure that this enzyme was working in this direction. Then we went in the reverse direction, that is from 3-hydroxybutyryl CoA to crotonyl CoA. This is another chromatogram. Here it's the black line, that is standard crotonyl CoA here, and then blue line, that is standard 3-hydroxybutyryl CoA. Then pink reaction, that is a control reaction without enzyme. And here you can see that it is on the position of crotonyl CoA, there is no pink peak, or not even in the surrounding of this crotonyl CoA. And then there is the green line, which is the reaction, and here is the appearance of peak. In HPLC, it's very sensitive analysis, and all peaks should overlap with their standard ones. But here is the displacement peak, and now we are thinking either it's crocodile CoA or it's different product. We are working on it also. But then we did another assay that instead of the purified protein, we took the whole protein isolate of the pyrobaculum, and we did the same assay, and here we got the peak that is really matching with the crotonyl CoA. But we are confused, either this peak on its position was due to really this PKL1306, which is the gene in our operon, or it's some other hydratase in the whole protein set, or it's because of all proteins of the operon are present in lysate, so we got this peak on the correct position. Next gene was acyl-CoA dehydrogenase, which can convert this butyryl-CoA to crotonyl CoA. And here we also analyze the products with HPLC. Here we have the black standard line, that is butyryl-CoA. Here you can see. And then orange standard line, that is crotonyl-CoA. And the green control, which has butyryl CoA but not enzyme. So here we can see butyryl CoA on the green line, but here we cannot see any peak on the crotonyl CoA. But in the reaction, this butyryl CoA was consumed, so peak disappeared, but a new peak appeared here. So it was the crotonyl CoA. This FAD, PMS, these are the reaction participant for this enzyme. But next thing is that, okay, this is 1306, this is 1308. Either the substrate of one enzyme could be, the product of one enzyme could be the substrate of the next enzyme or not. So what we did, we started with the butyryl CoA, we added this 1308, but we did not added this enzyme, and we were looking for this 3-hydroxybutyryl A product. So we had multiple reactions, few for the standards, that is because now we need three standards: butyryl CoA, crotonyl CoA, and 3-hydroxybutyryl CoA. Here is butyryl CoA, crotonyl CoA, and 3-hydroxybutyryl CoA. So the black is butyryl CoA standard, sky blue is 3-hydroxybutyryl CoA standard, and blue is the control reaction which does not have this 1308, but it has this 1306. But in this reaction, we could not find any 3-hydroxybutyryl CoA. And then we had the reaction with 1308. And then we had the orange line, which is uh, uh, the reaction with both butyryl CoA and crotonyl CoA. So in the reaction where we have just 1308, that is acyl CoA dehydrogenase, we had this peak, but there was no butyryl, no or almost very less 3-hydroxybutyryl CoA, which might be some error. And once we had, but I don't think so. Okay, and then we have the orange line. Here you can see, here the consumption of this intermediate product was highly improved, and there was the product at the 3-hydroxybutyryl CoA. So we were sure that, okay, these enzymes are interacting with each other. Then we have the assay for the acetoacetyl-CoA reductase and conversion of acetoacetyl-CoA to 3-hydroxybutyryl-CoA. So the green line is acetoacetyl-CoA, and then black line is 3-hydroxybutyryl-CoA. Pink line is control, where we have acetoacetyl-CoA but no enzyme. So no consumption of this pink peak, and appearance of product is neither seen on the this end. But in the blue reaction, we can see that this acetoacetyl-CoA peak was reduced, and then the appearance of 3-hydroxybutyryl-CoA. So now things were making bit sense. And then we went on the other way from acetoacetyl-CoA to crotonyl-CoA. Multiple reactions, but here I will just show you that here again we got the crotonyl-CoA, this product, something from this product, without adding this intermediate. But again, we got with displaced peak. And then we had another enzyme that is uh, 3-hydroxybutyryl-CoA epimerase, or it could be the DOF protein. So when we added this protein, there is difference in the consumption of this acetoacetyl, slight difference, acetoacetyl-CoA, and then the slight difference in production of crotonyl-CoA. So we knew that it has some role, but we could not identify yet. We are working on it. So up till here, we can see that we started from butyryl CoA and we reached to 3-hydroxybutyryl CoA, and then we started from 3-hydroxybutyryl CoA and we went in reverse direction. And I have not talked about this enzyme because this enzyme is giving such much tough time. It's not been expressing, then it's expressed as a nucleon body, then we expressed it. Now we are searching for its activity. So now this is the question that how we can go from this to this, and why this thing will work, and what is the role of esterase here? Is it really working with this? And either by the involvement of this alcohol dehydrogenase, it can be used for the biobutanol synthesis or not. So the outlook of this project is to characterize the other player of beta oxidation from Pyrobaculum caladifontis and to link their activities and check their activities inside the cell. And if we could really identify a functional fatty acid pathway, and then we will try to solve out really, it could be the last common universal ancestor which has the both functional isoprenoid biosynthesis and fatty acid biosynthesis or fatty acid breakdown. So this is all from my side. I tried it to make it simple, but still. So thank you so much for your attention. So if you have any question, just let me know.
Thank you so much, Dr. Vella, for such a wonderful Just a part of your study, I believe. So, anyone
Sorry,
from students
okay,
and
I hope no question.
Assalamualaikum, can I talk?
Yeah, yeah.
Okay. Dear Raja, it's really wonderful. It's really, really wonderful. And one thing that I surely believe that the science is interlinked. There is not biology, not botany, not zoology, and not chemistry. They are they all come in under single umbrella and they are connected. And you have proved by talking about the evolution, and I'm so happy. Your work is marvelous. Uh, I'm
Thank you.
inspired,
Thank you so much.
and I'm proud of you, my dear. And happy
Thank
Women's
you.
Day. You,
Thank you.
the
Same.
women and the scientists of like you make all of us proud. Thank you, and thank you,
Thank you.
Ivester, for introducing such an enthusiastic and wonderful scientist among this community. Thank you.
Sure. Thank you so much.
thank you so much, Dr. Raza, for your time and for giving us such wonderful
I'm sorry, I could not hear you.
um, am I audible to other audience?
I don't know because your volume is so low.
Okay. Am I audible now?
Yeah, yeah.
I was saying thank you so much, Dr. Raza, for your nice and wonderful talk, sharing information about your project and the wonderful journey of how you reached here, starting from your VS now reaching here,
Yeah,
Germany, with
yeah.
such difficult hurdles and tasks you did perform and you achieve your goal with your determination.
Okay.
Is really an inspiration for all the girls who are pursuing their careers in the field of molecular study, genetics, biochemistry. So I think you are an inspiration for all of them. Thank you so much
Thank
for
you
your
so
time.
thank you so much. The most funny thing is that it's quite morning here, and I think I requested you to make my presentation on Friday because of I had some plans on Thursday, and unfortunately, the plans were moved to Friday. And the most funny thing is that with this presentation, I am right now cooking halim and plow also on the side project. So women are always in the kitchen. It doesn't matter they do science or not. Okay. Thank you so much.
And actually, this is all about Dr. Raza Ashraf, as she was telling us about her journey. And same thing happened to you for today, that the things got merged as you planned and as nature has planned for you. So you always get the same targets. Thank you so
Yeah.
much, Dr.
Thank you so much. Thank you so much
So
for
for
this.
moving ahead towards the second speaker of today's wonderful session for Women in Science, I welcome Dr. Bushra Nisar Khan, a very esteemed speaker for today, um, for sharing her knowledge, for sharing her journey at this forum. So, Dr. Bushra, we welcome you all here for the life journey, your experiences, and your talk. Dr. Bushra, I think you have lost your connection for a while. Let me wait for her to join again. Dr. Bushra will be joining us in a while as she has some connection problem. Anyone from the audience, if want to say something about the special day today for at Avista Forum, you want to share your few words about women in science, you are welcome to say if anyone from the audience like to share.
Oh, sorry. I'm so sorry. Am I audible?
Yes, you are audible.
Thank you. I'm so sorry. I'm really so sorry. I just lost the connection, and that is why I am unable to talk to you. So I'm so glad and I'm so thankful to Ivister and Dr. Amara Dar for providing me an opportunity to talk at that very special day of the women to celebrate the success, the deeds, and their work around the globe. Thank you very much. Um, uh, so I saw the video Amara shared with me for the iVistor, and I saw the whole thing is dealing especially with uh, the science and technology. Uh, but here I want to introduce a new window of the science, which is dealing with the animal sciences. And animal sciences is again in a whole field which deal with the which needed modern science, technology, and all. And we start from here.
Dr. Bushra, can you share your screen?
I'm sharing a small video. Is it visible over here?
We cannot see your screen.
No.
Dr. Raza, are you still in the meeting? Then you can please check your screen share if you have not stopped it yet.
Ah, I think I'm unable to show you what I have uploaded. And now, is this screen is clear to all of you or not? I'm sharing in presentation. Is it visible? Okay. Actually, I am thinking Raza
Um,
screen is open. That is why.
Dr. Bushra, now your screen is, will be visible. Please share your
Okay.
screen.
Okay. Thank you. Is it visible now?
Uh, yes, it is.
No. Hmm. Okay. Here I am showing you the pictures of all those animals to whom I spent 8 years of my life. They are the beam of inspiration. They introduced me the love of nature in myself. Um, you people can see the picture of Susie over here, the childhood friend of many of us. Okay, and here I have started my journey at Lahore Zoo, mm, because due to the change in the problem in sharing of the screen, I totally lost. So I try to come back to you people again with new enthusiasm. Okay, in 2004, when I was doing my MPhil, my many of the friends who have done with their MSc are started their jobs. But I was the one, and many of the other one who are looking for the job. And I was thinking maybe I would be able to get a job of a school teacher and all like the other girls after doing their MSc and BSc are doing. So I was also thinking I was dropping my CVs in different schools and organizations so I would be able to get a job. Suddenly, one day, at that time, the reading of the newspaper was very common in people home. My sister, my elder sister, Zebunisa, so I cannot forget to mention her name over here because if she did not see that advertisement, she will not inform me, and I was unable to share this lecture or my success story with you people. She said, in Lahore Zoo, there is a post of education officer is open, and their requirement is MSc Zoology. Are you interested to join? I said no. How can I work in zoo? Zoo is not the place to work. Here the animal are present. People will make joke of mine if I join the Lahore Zoo. She said, no, it's very golden opportunity. You should. But on her emphasize, I have submitted my application over there. But from my inner side, I was not thinking that I should join the Lahore Zoo. But finally, as Allah has decided me to over there, I joined Lahore Zoo, a very new and different place, an area which I have visited many times during my childhood, during my studies, being a zoologist, for a visit, for a tour, and just for the fun. And the only memory and only the famous thing that is about the zoo is the order of the animal. People avoid to going zoo, especially the elder one, because zoo is very much there are lots of unbearable order in the zoo. At zoo I realize why this order is there and why zoos are important in our life. Initially, it was really very difficult because in the Lahore Zoo at that time was 132 years old. It's a very old zoo, and it was an old donated by Lala Mahendra Ram, an Indian who got this small every type place from Queen Elizabeth. Okay, and since then there was not single lady working at the zoo. And I was the young, just the student of my MPhil zoology, and internally I was not liking it. But gradually, the love of the animal and the nature, I also myself want to be a cancer biologist, a molecular scientist. This was my dream, and I always trying that one day will come, then I will leave the zoo and join this advanced zoology or advanced biological techniques. But gradually, I realize the place where I am is the birthplace of the earth, where I identify my link with Allah subhanahu wa ta'ala. Saw the organism in very close connection with the nature. And finally, when I used to visit the zoo, many of the animal become my friend, like chimpanzee family, lion family, tigers family, elephants one. They recognize me, they wait for me. I visited them and I talked to them, but the people or the male community of the zoo were not so kind to me. Only the director of the zoo is so nice and so cooperative. He is the one due to which I didn't leave the zoo, and I was remained the part of Lahore Zoo for 8 years, for almost 6 years, from 2004 to 2010, before joining my Punjab University. The other people tried to create many hurdle. They felt that I am a young lady and I'm afraid of the many things. They create problems. They always used to tell that there is no need of any kind of education and research officer. She is useless. Her post is useless. And the zoo is the place just to work for the male community. Women are not required over here, but only my director, who always gave me strength and courage, that no, you should go ahead and identify your ways. No structure of education over there. I don't know what I have to do. After roaming for 3 months, I realized that what difference I can do over there. I started communicating with the people. Okay, I started communicating with the students. I started delivering the education or the knowledge, the pain, happiness of the zoo, and the importance of the zoo to people, that the zoo are not only meant for the recreation. For the International Union for the Conservation and the VASA World Association of Zoo and Aquarium, the first and the foremost goal of the modern zoo is the conservation, research, education, and lastly, the recreation. Then I realized people become familiar. School people started doing zoo visit in better way. Many of the veterinarian, many of the students from the botany, zoology people started do their research work over there. I have introduced many education activities over there, zoo education program, zoo internship program, zoo animal adoption program, zoo animal activities, and different kind of celebration like the World Environment Day, World Wildlife Day, World Wetland Days over there, World Vulture Day, Ornithological Day. Gradually, the face of the zoo just to come, eat, drink, throw the stone on the animal was finished, and people are realizing that this is the place where we can enjoy and we can interact with the nature. At that point, there was no time. I don't have an office, I don't have a good place to sit. But gradually things come to me. I arranged computer on my own from donation. I arranged camera. I arranged printer. Nobody was there to help me out. They because they were as lastly the Sadaf Shah told yesterday that washroom was the issue. Yes, it was issue over there. I'm feeling very insecurity, but gradually things turn out in good shape. People started recognizing me, the role, and my job over there. I was becoming happy. I became the media correspondent. During this tenure, I became the member of IUCN education group. I am the member of South Asian Zoological Association for Regional Cooperation. I have visited Nepal, Sri Lanka, Thailand, and many other places to visit the world class zoo, America, Japan, Nepal, India, and I have seen the zoo and got many training for zoo management and all zoo nutrition, animal pairing bonding. Then at 2010, I have realized that it is time to do some lab level research to support this speechless creature. Then I joined Punjab University and I came to Center for Undergraduate Study and where I met my lovely friend Amara Dar. I have started my work over there. And one thing that was always pinching me and making me realize that I have to pay back to zoo and I have to pay back to all those speechless creatures that that make me what I am today. And here I cannot forget the sacrifice and the hard work of my mother. She was there at every step. She helped me, she dropped me, she picked me, my sisters. So, um, everywhere the situation is not so worse. Some families are so supportive. If I am today, I am Dr. Bushra Nisar Khan, then behind the screen there are many male and female, my friends who supported me and who encouraged me and who make me the Bushra Nisar Khan. So what I realized, the main thing, the very important factor at the zoo that destroying or that is disturbing the zoo animal is the stress, the captivity stress, the stress of same kind of food, the stress of same kind of habitat, substrate, same environment, some same visitor vandalism, torture, and all. They should be free of this torture. And then to release this torture and to release this stress, I decided to help them out by my research work, and I started work on animal welfare to reduce their stress by different kind of the therapy animal enrichment program. Second step that came into my way is that how can I do the sampling, because in wild animal getting the blood samples and capturing them is another stress. If we capture them, I witnessed this issue when zoo herbivores were suffering from the tuberculosis. The people from when VRI people came for their vaccination, even during the vaccination process, when they tried to capture or vaccinated them, they got injured, and even their heart was busted, and they died just due to capturing stress. So I have to look out the procedure or the method that is totally stress free. Luckily, in 2005, I have visited I have presented my research article in American Association of Zookeeper. There, I become familiar the process of non-invasive method of cortisol estimation. A very young veterinarian in St. Louis Zoo shared her presentation to estimate the cortisol from the fecal sample of the wild animal. And it was so easy, without any stress. We just picked the feces and estimate their fecals with the help, we estimate their cortisol enzyme concentration with the help of the ELISA technique. I was happy because I got the answer of my question. When I came back to Pakistan, I was finding out the way that in our medical laboratories, the estimation of the cortisol concentration in blood was very common, but the estimation of the cortisol concentration from feces, from urine, from feeder was not that much common. After helping, after connecting the lady in America, I came to know the way by which we can estimate the ELISA concentration in zoo animals by just picking their feces sample. I established the ELISA estimation lab in my conservation biology lab, Institute of Zoology, and I have started uh, work over there. So today, I am just sharing a small piece of my research work on the estimation of ELISA concentration in zoo animal and the use of the aromatherapy. Aromatherapy is a very effective therapy around the globe. The use of the phytochemical to reduce the stress for sleeping enhancement. You people have seen the many product of the Johnson's baby or the baby product. They they have in their night products the lavender oil massage, especially for the newborns or the baby. The simple massage with the lavender can make them fall them sleep, and it's easy. And even the massage of some essential oil I have seen in Thailand. The massage is very common. Even on the footpath, the people were doing the massage to get rid of many kind of their remedies, their problem, and one of them is the stress. So here I started my presentation. Bismillahir Rahmanir Rahim. And the topic, why it's not moving? Okay. It's zoo animal welfare in 21st century. Zoo. Zoo and the interacting with the animal is the part of human history since ages. Okay, people used to keep the animal, to interact the animal, to fun, to game, to hunt for medicine, and many to cloth and many more things. But in 19th century, zoo were old-fashioned menageries, where the animal were put in very bad cages without zero animal welfare. Gradually, these concepts were changing, living in a naturalistic cabinet to the it moved toward the zoological parks. Here, the main theme is ecology or living museum. The animal were little bit in a better condition, but not that much that they should be close to their natural environmental condition. And in 21st century, zoo are the conservation centers. Environmental resources, environmental ecosystem, survival of the species and the species of the concern of the endangered species, and many more. The exhibits should be look like that they are the animal are in jungle or very close to nature. These are the 19th century old fashioned menageries when animals were kept it into small cages like the circuses. And these are the 21st century. The picture where my cursor is the moving is the picture of Lahore Zoo Safari. We can't say, although we are not up to the mark, but we can't say that we are totally in that condition of the zoo that totally against the animal welfare. The main reason is the resources, skilled person, and other thing. No doubt, with gradual in change, we are creating and we will create wonder in animal science and animal conservation. Same the way the new upgradation plan of Lahore Zoo and Safari is undergo these days. And after the zoo and the safari will open, you people will realize that they are much better than the previous zoo, and the animal are more happy and close to their environmental condition. So here I am presenting how can we estimate the cortisol concentration from the fecal by adopting a non-invasive method and after the administration of the short-term aromatherapy. I have given you the all introduction. What are the essential oil and what they are doing? This is clear to all of you. And here I am telling you what kind of oil I have used. I have used four kind of the oil: lavender, eucalyptus, lemon, and the peppermint oil. Four is short-term aromatherapy, and after aromatherapy, we picked the fecal sample. My very brilliant and good student has done this work under my supervision. First of all, we picked the sample before the administration of the aromatherapy. Then we picked the sample after administration of the aromatherapy, and then after the post enrichment, just to check that which oil is the more effective one, for how long the period of effectiveness, and when the second episode of aroma should be introduced into the cages of the animal. Okay, we online purchase these oils, and these are the oils. Hafiz G Oil Company was that. Okay, and the animal under study were the carnivores of the zoo. These carnivores were housed in a very small cat section, but now when you will visit the zoo, this cat section is converted into the animal, the bird colony. Okay, so it is very small. The cages were so pathetic, animal were so tense, especially the Asiatic wolf was in very bad condition. The Asiatic lion, common leopard, stapid hyena, jungle cat, and Asiatic wolf were our animal of study. And this table is presenting you their status and their cage side. They are, as I have told you, in here you can see the outdoor cage measurements and their status. All of them were like some were critically endangered, vulnerable, near threatened, and some were least concerned. And in this same slide, I have shown you their distribution and the way they are keeping at zoo. This was a small ethogram. Whenever we study the organism behavior, it is very necessary to design an ethogram. An ethogram is telling us the different kind of the behavior like the stereotypical behavior or the normal behavior or the abnormal behavior. For example, of the abnormal behavior, if a carnivore like the lion, tiger, or any of them is moving in their cage in this way that it's making a figure of 8, it is the clear mark that this animal is under severe condition of stress. Okay, so in upcoming slide, I will show you the Asiatic wolf is moving just in the pattern of 8 and showing his condition of the stress. But I am also happy to share over here this aromatherapy help us to reduce their cortisol concentration estimated by the ELISA, and even their physical behavior was also shown this thing. This here you can see the cage. You can understand the pathetic, how pathetic it is, the condition. It is the cage of the wolf, jungle cat, a very small cat, and a very bad, very small cage and a very badly managed cage. This is a common leopard. Uh, I am very sad to share that these common leopard are gradually declining in our Himalayan or Mari area, Margla Hills. Reason is that due to human leopard interaction, leopards came down into human dwelling and killed their livestock. And ultimately, what the people are doing, they are constantly killing this very important, magnificent, and ecologically important carnivore day by day. Only thing which we have to do, we have to save the livestock or the people life at one hand, and at other hand we have to save this common leopard as well, because they are the pride of Pakistan. They are the important carnivore fauna of the Pakistan. The strategies or the concerned department should look the way that how can we save its habitat, that it should not come down into human dwelling, and how people should be aware that they should not put their cows, their goat in the habitat of the tiger, in the common leopard. And one more thing, its name is Guldar in Urdu. Guldar, Gul, an organism जिसके जिस्म पे फूल खिले हुए हैं. If you people can see, its body have a roseate pattern. There is a pattern in center, a light brown pattern, and around the pattern there are a 5 prominent pattern in kind of the petals. So that is why इसको हम उर्दू में गुलदार कहते हैं, एन ऑर्गेनिज्म जिसके जिस्म पे फूल खिले हुए हैं, और ये गुलदार की स्किन है जो उसको सबसे ज्यादा हंटिंग करवाती है. देन वी हैव द एशियाटिक लाइन. दिस इज द स्ट्राइप्ड हाइना जिसको उर्दू में हम लगड़ बगड़ कहते हैं. एंड these are the phases of the enrichment: pre-enrichment, enrichment, and the post-enrichment. These were the phases. Okay, and here we are introducing the essential oil on the wood log present in their cages. Where the available space was there, where we were thinking, because prior to introduction of the oil we have visited or we observed the animal behavior, that where they used to come and which thing of the cage they touched there first. Okay, and we observe two things in their behavior. One is the frequency, and second is the duration of the abnormal behavior. These are the ELISA, and what are the result? If you see at the result, the cortisol concentration of the ELISA in Asiatic wolf, you can see there is a prominent changes in their behavior or cortisol concentration. Please monitor its pattern of 8-like movement. This movement is the clear sign that it is in stress. It is suffering from severe stress, and during its movement, it is making an 8. All the time restless, always moving in this small cage from right to left, left to right. Again, it is the cortisol estimation of the cat. She is smelling the aroma we sprayed before her entry into the open area. She is feeling the change in the cage is there. Common leopard. The leopard is also sniffing. An Asiatic lion and striped hyena. The hyena also smelled and feel the difference. This is an enrichment, the aromatherapy linked enrichment, where animal can identify any change in their cage, any change that is different from their routine work and movement that make them happy, that help to reduce their stress, that make attraction in their cages. We felt that these organisms, these all animal feel, smell, and like the smell, and even after 7 days of the enrichment phase, at the 8 days when there was no oil spray, they were looking here and there just to find out the where is the oil and where is that new thing that was introduced in their cage. Summary: among the oil we find the lavender is at number one, eucalyptus at second two, peppermint, and lastly we felt the lemon. So among them, the most effective one, the lavender and the lemon, was the last least effective to reduce their stress and their and just to reduce their behavior they presenting by the we observe over there. So it's also observed that after 10 days or the during the post enrichment time, after 10 days, first we estimate the cortisol at prior to introduction of the aromatherapy, then after aromatherapy, after 7 days, and after the aromatherapy, like the post enrichment phase, for that after 10 days we again estimate the cortisol concentration, and we found that it was again elevated. So it was obvious from this finding that it is for the short time. So the zoo management, I have recommended, we recommend the proper sizes of the enclosure, proper vegetation cover, pairing of the animal, captive habitat should be close to a natural habitat, and proper enrichment regular plan. I met the Director General of Wildlife, Zoo Director, Zoo Veterinarian to share my knowledge, and I told them that these are our finding. If you people do some little enrichment, so do some little effort, their cage will be attractive. They will be happy, and we and you will achieve the goal of animal welfare. Thank you very much. This is all about my side, and the house is open for your questions.
And that is, I think, is a great contribution with your science education that you are uh, doing to the community, and you have done already so many wonderful tasks, and you have won awards and everything like that. So, I welcome audience if they want to ask something about the research work or the journey of Dr. Bushra Sahan.
Hello.
Hello.
Hello. Thank you so much for such a cute presentation.
Thank you.
That
Thank you.
Zool has always been a place of interest for me, and not only for me, for my mother as well. As soon as I come back to, although she is 70 plus, but still she loves to go to the zoo. And
Next time we
me,
will visit together. Next time we will
yeah,
visit together, zoo and safari, inshallah. And we will
yeah,
also try to cooperate with you from anything which we can do for this speechless creature. They make
yeah,
us happy. They are source of inspiration. They are a childhood memory. They need our attraction, love. We'll do
yeah,
something for them.
yeah. And I remember that whenever we go, we always heard from people, oh, this wolf is mad. Because I think the wolf is alone. It does not have partner there, and
Yes.
it always moving from left to right, and
Yes, yes.
Whenever you go, so you know the same movement, and everyone's saying, "Oh, this wolf is mad. This wolf is mad." Whenever you come, it's but now I really know why he is mad. He is not mad because of the stress.
Yes,
And
yes.
yeah, and also I think for the lions, they are sometimes they are also moving, and it's very nice. I always love lions, and I always go there to see lions and hippopotamus.
Hmm.
Thank you so much. And really, I will
Welcome.
contact you for next visit to
Inshallah, inshallah, very soon. Inshallah. The next time I Wester will be in the horse safari at Tiger Lion Safari, inshallah.
Yeah, okay. Thank you. Yeah, but I'm really happy because we have been hearing this all aromatherapies for human being in
Hmm.
very expensive spas, and especially for this lavender oil that you keep some. But it's
Hmm.
good that if they can be used for the animal stress elevation as well.
Yes, because you have seen
Yeah.
people who are in stress, they used to move here and there. Or
Yeah.
zoo are earning a lot from this animal. So why should they can spend? At that time, the DG was ready, but soon after this research, the zoo have started its upgradation plan. And when animal will back in zoo, I will definitely pursue this, and I myself will try to introduce. Even if we get some sponsorship, some person who are selling good oil, they
Mhm.
can donate this oil to animal welfare.
But this is very unfortunate because I remember once I went to Hunza and I saw so much lavender in the wild, and
Mm-hmm.
nobody in Pakistan really bothering to harvest those lavender and everything. And we are always importing this lavender oil from
That
like,
really well.
yeah.
Next time we should also think about that we should harvest our own lavender and produce oil. As not for the animal use. Many
Yeah.
people in our community need this kind of favor. They should also
Yeah.
people stress free.
Yeah. Okay. Thank you. And
Welcome,
I
welcome.
was cooking and I was hearing your presentation.
Thank you very
So,
much.
By
I wish
chance we
I
had
wish
yeah, we
I
had some one dish party from Asian students in our group today. So
that's great. I wish me and Amara can eat.
uh, maybe when once I back to Pakistan, then I can invite
Inshallah,
you.
inshallah, inshallah.
Okay. With Amara, I
Thank
have very long time relationship since my first day of BS
you.
in Punjab University.
Hmm.
Okay, thank you.
Thank you. Welcome.
I'm well thankful behalf of my team. I really feel the pleasure of having you with us and sharing your experience, sharing your research, sharing your knowledge, and keeping all the other females inspired of your work and your personalities and your journey. So, what we believe is that hurdles and you can say problems always keep you elevated if you have some goals in front of you and you want to achieve that goal.
Sure.
So, for the women in science or women in even the women in kitchen, the thing that we have in our mind is that we want to achieve something. So women are a very wonderful creature of Allah, and God has blessed them with so many qualities and so many abilities, keeping them deprived of achieving their goals or keeping them deprived of educational facilities due to some societal constraints or everything. We should work over it, and we should not bound the females not come out of the box approach. As we have seen from the presentation of Dr. Bushra Nisar Khan, that when we keep the living organism away from the natural environment, how restless they become and how short-lived they become. So for same is for the female, for the women, Allah has blessed them with every ability and every potential to work in each and every field of the science. So STEM field now and now onwards can never be the strong pillar in development of the globe if we exclude women out of it. So
Sure.
happy Women's Day to all the ladies, all the female, all the girls who are on this path of getting education in STEM, on working, or they are working in STEM and they are contributing a lot in STEM. Thank you, and happy Women's Day to all the females from IWD, Pakistan. Thank you
Thank you
so
very
much.
much. Thank you very much, Amara, and thank you very much, iVistor, for this opportunity. Happy Women's Day to all women in iVistor, and Happy Women's Day to all my student, my daughter, and all of us, wherever, because even kitchen is it again in science. So everywhere women are working, they are creating wonder. Thank you very much.
Thank you so much. So,